Well, hi and good morning, everybody. We are really delighted that you're able to join us. Welcome to the third in our Life Course Research Visual Toolkit webinar series. My name is Sherry Barkin. I am at Vanderbilt University Medical Center. I'm delighted to be part of this executive team to bring these opportunities to you. I'm also joined by my other leads for this work, and that is Dr. Heidi Hansen and Dr. Rick Kaskell. So, as a reminder, the Life Course Enterprise Committee, with support from Utah's and Vanderbilt University Medical Center's CTSAs and the Maternal Child Health Bureau's Life Course Intervention Research Network, we have created both synchronous and asynchronous tools to build our knowledge and application of life course research. We are delighted to introduce our third in this series. There will be a total of 6 of them. Our topic today is utilizing data science resources to prepare and package integrated data sets. Here is the way that we're setting all of our webinars up. The first is that we have a TED-like talk. That's to give you a broad overview of the topic at hand for the day. After today's TED talk, we're also going to have 5 minutes of questions. As you have a question come to mind, please use on the bottom of your screen the Q&A box for your questions. And as you have them, please post them. We do have a moderator today, Jomi Ramsey. She'll review all of the questions and we'll make sure that we have a chance to ask them with our 5-minute period of time after our first TED talk. The TED talk is then followed by 2 how-to talks. You all have requested from us how we take all of this interesting theory and we put it into action. We're really fortunate to have some incredible speakers who will give you some specific examples of how to put this into action for you as you conduct life course research. So we're going to get started with our first TED Talk. I am delighted to introduce Dr. Joshua Denny today. He's the CEO of NIH's All of Us Research Program. He's an adjunct professor of biomedical informatics at Vanderbilt University Medical Center. His research interests include natural language processing, accurate phenotype identification from electronic medical record data, and using the electronic medical record data to discover genome-phenome associations. This allows us to better understand disease and drug response, including the development of what is now known as the phenome-wide association. For many of you, you know that as PheWAS. He really has been such an incredible innovator in the field, and we're delighted to introduce him today for our TED Talk as we begin our discussion of utilizing data science resources to prepare and package integrated data sets. Dr. Denny, thank you.
Well, it is a pleasure to come to everyone virtually today. One of the powers of we have all explored virtualization, and I think it has become a powerful tool. It is a pleasure to share some of our experiences with All of Us Research Program, but also more broadly, I'm going to talk about precision medicine and some of the journey that we have here. First, I think about, what is the problem we're trying to solve? And the first is, I think of this as the differential disease risk that patients have. We all know that we desire a healthy health trajectory, but individuals and all of us over time will be exposed to disease, and the disease can have quite a variable disease course. And some of these diseases, we have centuries of history on how we have named them, but they represent potentially different entities. And the same is true for how we handle drug response. Obviously, the goal is to have the drug work the first time. Sometimes we also have toxicity to it. Sometimes the drug does nothing, and sometimes it's only toxic. And we don't have very good methods of predicting much of this today. And our goal with precision medicine is to try to get better at this, both with disease understanding and therapeutic delivery. Many of us are familiar with the Framingham Heart Study, really one of the many transformational studies out there that looked at a small number of individuals, in today's standards, about 5,000 individuals that were studied longitudinally. And through this, discovered things such as the impact of high cholesterol and high blood pressure on cardiovascular disease, stroke, the fact that smoking was harmful, that exercise is beneficial. And, concomitantly with these kinds of observational cohorts, we've seen a dramatic reduction in cardiovascular disease over time. Similar curves have been seen for cancer and other diseases. Yet most of this tackles common disease and maybe doesn't necessarily think about the individual variation that we all experience. And similarly, nothing similar has been really seen for how we can better target therapeutics other than understanding the root of biology of diseases. One of our challenges is whether or not we can have a similar experience with precision medicine, and I use this synonymously with personalized medicine in this case, over the next 40 years. Can we really transform healthcare to be more precise, to better understand the biology of our diseases, and better deliver rational therapeutics to our population? There has been an amazing growth with genome-wide association studies, sequencing studies, and other use of genetics to understand better disease risk. Most of these studies have looked at occurrence of a given disease. One of the powers of having rich electronic health record data or longitudinal information is something akin to what the Framingham Heart Study did, which is, in this example, looking at people who are put on a cholesterol-lowering medication, for instance, to prevent heart disease, but yet still have a heart attack. How we use the longitudinal medical record with up to a decade or more of follow-up on some individuals to find that LPA variants were an independent risk factor, which actually predict levels of lipoprotein A of having either a secondary or primary cardiovascular event despite being on cholesterol-lowering medication. And indeed, that was something we found true even of those that had ideal LDL levels of 70 or below. But one of the powers of having a really rich phenotype set is you can do something like the phenome-wide association study. So what we did is then we took this variant and said, well, gosh, if we targeted this variant, what might we see? So we did a phenome-wide association study on that locus, and what we found is, fortunately, coronary atherosclerosis and other similar conditions were the highest ranked conditions, and a number of heart conditions showed up. That is suggestive that targeting this locus for anti-cardiovascular disease therapeutic would likely not have off-target effects. And using the electronic health record, using genetics in this way, is a tool that is being used within the pharmaceutical industry and other conditions to help us repurpose medications and similar sorts of drug discovery events and de-risking as well. Having these kinds of data, let us look across the data at scale. This is a study that, we've had about 5,000 people in the Framingham Heart Study. Now we're looking at about 40,000 individuals. Really actually a small set if you think about some of the really large biobanks that exist today. This study is looking at HLA markers and all their disease associations. And what we can see with different HLA markers are a number of autoimmune diseases that are associated with a number of these different markers. And we can see different classes of associations, in different numbers of them with different HLA regions. This has an added power of not only just sort of replicating that approximately 40 years of HLA investigation in one study with over 100 associations, some of which were novel. It also allows us to look at whether one HLA variant that looks like in two independent studies may be associated with two different autoimmune diseases is indeed associated with that in a pleiotropic sense, or whether they are independent, whether truly independent association, or having risk of diabetes leads to further risk of rheumatoid arthritis, maybe potentially later in time. And indeed, we were able to see a lot of those truly pleiotropic associations. There's also impacts on rare disease. We're all familiar with the growing list of Mendelian disease, with over 6,000 of them identified, and really the count and the mechanisms being driven more each year than the year prior. But we can also look at clusters of symptoms if we have deep phenotyping of individuals to find people who may not have been diagnosed, or to help us interpret genetic variants that we don't know information about. So this is an example of using the data in electronic health record. We're going to consider cystic fibrosis in a number of scenarios here, such as bronchiectasis, pancreatitis, asthma, failure to thrive. These are all components of cystic fibrosis. And so each of these have a different, I'll say, evoking strength for the given disease. Bronchiectasis is much rarer than asthma, for instance. And so, what we did is we weighted these based on their rarity in the electronic health record across all patients, such as bronchiectasis has a different weight than asthma. And then, that creates an aggregate score for an individual. So this top individual has a higher score than the bottom individual. And, someone that doesn't have any mansion symptoms, would have a score of 0. And so what we found is, this allows us to differentiate highly statistically, given condition like cystic fibrosis from people who don't have cystic fibrosis, purely by using the symptoms they have in the electronic health record. In fact, when we went back in our population, we found one individual who was newly diagnosed at age 80, who had had symptoms for decades, for instance, of cystic fibrosis. That's obviously an extreme case of someone that had obviously hand-over conditions and had less penetrant, less severe variants, causing her cystic fibrosis. But it is something that reveals the power of these sorts of approaches. And this is, it worked over many different Mendelian diseases and is helping us interpret the pathogenicity of variants. Let me give another example here. This is an individual presented to our undiagnosed disease network situation. She first presented with hearing loss and numbness. She developed an auditory neuropathic syndrome, blurry vision. It continues to progress and has muscle spasms, neuropathy, is confined to a wheelchair. She ends up getting a feeding tube and eventually is intubated and comes to the undiagnosed disease network through a combination of history, physical exam, deep exploration, and whole genome sequencing. She was diagnosed with riboflavin deficiency and was put on a very high dose riboflavin and regained a lot of her prior function. She regains a lot of sensation. She's able to, of course, come off the respirator and is able to walk without a walker. And if you go back and look at her phenotype risk score for riboflavin deficiency, she was 14 standard deviations away from what would be expected. And so, highly statistically derived towards this as a potential. And it gives a sense of how we can use both the phenotype and the genotype to help us interpret what's happening to an individual. However, we are all aware of the lack of diversity in science, and this has really been brought up with recent events in our nation that has reminded us and helped us look introspectively of the lack of diversity in science and certainly in genomics. This is also true of clinical trials. And if you look at where we are, while we're getting a little bit more diverse here, we still have a strong bias to those of European ancestry. And indeed, about 4% of all genome-wide association studies, according by this count, represent about a third of the U.S. population. So, the United States has an ability to contribute to the diversity of genetics that are done, and that really has really important impacts. We know about some of the clinical drivers here in the impacts in terms of normal calculations like, glomerular filtration rate, scores for recommending transplants. Many of these incorporate race or ethnicity, but maybe they shouldn't. And maybe we don't have nuanced understandings or incorporations of those as much. It makes our polygenic risk scores, which people are excited about how this could improve calculation of disease and disease risk and maybe treatment, but it doesn't work as well across different ancestries because we haven't measured them. And it impacts everyone because we don't interpret genetic variants correctly. We may say a genetic variant is pathogenic in someone because it was to study primarily Europeans, but in fact 27% of African Americans or Black individuals may have that variant. So clearly that wouldn't be pathogenic. So people are getting ICDs and other therapies wrongly without having the diversity, and there are many other impacts as well. So in January, I joined the NIH to lead the All of Us research program, which has many goals, but to propel precision medicine is its key, and really to value diversity across our country in so many different metrics, including race and ethnicity. We had our national launch in 2018. We believe participants are really partners in our program. It's a longitudinal study that's recontactable, but gathers lots of different kinds of information, including surveys, biospecimens, electronic health records, use of digital health technology, and in-person physical measures. Over 350,000 people have enrolled, including 270,000 that's contributed biospecimens, and over 50% of those individuals are of diverse race, ethnicity, and over 80% are underrepresented by medical research, which includes sexual and gender minorities. It includes people who live in rural locations, education, and socioeconomic status. Just like with other electronic health records, we have a wealth of longitudinal data on these individuals. This is our first data set that was released in May 2020 that you can now have access to through an application process. And if you look at the longitudinality, we have data in some cases back to 1980 on millions of individual diagnoses. The surveys, of course, start once enrollment happened, but over 100 million labs and measurements, over 18 million drug exposures, and these sorts of things create a powerful data set over time. This is just one example of a study. We did a series of demonstration projects which are available, and you can actually reuse these analyses in our cloud-based data model and research platform and come in and see this and use this. In this case, we're just looking at people who have been diagnosed with depression and what their medications are. And if you look at individuals who are self-identify as white and people who self-identify as race ethnicities other than white, you can see there's actually a different pattern of primary treatments. So both groups, SSRIs, are the most common treatment started. But second, an individual self-identifying white would be bupropion, and you see second in non-white individuals would be trazodone, and then TCAs, whereas TCAs, tricyclic antidepressants, are fourth in individuals identifying as white. This is just highlighting a difference. There's so many more that can be explored. We've done a number of other demonstration projects as well in this process. So, as I think about this, all of us represents one study with a goal of recruiting at least 1 million individuals who will share this kind of information and be recontactable, with more than 50% diversity. But really, we need many more in global alliances and global data sharing mechanisms to allow us to investigate and really unlock the power of personalized medicine and discover and really counter some of the effects of underrepresentation in clinical medicine and biomedical research that we've seen so far. So these are challenges that you'll hear some about with our next speakers, and great opportunities as we think about the path forward. So I really appreciate the chance to talk with you, and I would be happy to take a few questions.
If you have a question, please put it in the question and answer box, and then Jomi, our moderator, will review it. Jomi, did you want to review some of those questions with Dr. Denny?
Yeah, so we have one question right now, so we'll get started with that one. So first question is, how will integrated data sets include the social and physical environment as contributors to the phenome?
That's a great question. Thank you for asking that. We incorporate a number of different components, for instance, with all of us, which includes surveys and digital health technologies, and by asking things like location, we're able to link in data sets. So at this point, there are a few questions that get at some of those elements, but and we released a specific module around COVID-19 that gets at a few more of those questions around social determinants of health. We have a social determinants of health module that we're working on that follows many of the standards that have been established there that we intend to release, and then we'll link in other information sets for sort of physical environment, particulate matter exposure, those kinds of things that over time.
Thank you. So it looks like we're still waiting on a few more questions. So usually they're a little slow trickling in. If we want to maybe move on to the next speaker, and then if any more come up for you, we can ask later. Does that work?
This is Sherry. I do want to ask one more question before we move on, and Dr. Denny will continue to send you questions as they come in. Our visual toolkit is all about the life course. Can you talk a little bit about how is it that we are addressing the life course in these integrated data sets?
There was a couple of answers, I think, of that question, and it's a stimulating one from the perspective of both the technological perspective and data gathering and analysis perspective. One of the things I didn't talk about is right now all of us is limited for enrollment to those 18 and over. One of the things we're looking to do and build out over the next year or two is the ability to enroll pediatric populations, which allows us to think more of that. Longitudinal data capture, both through active and passive means, are part of that. It's something that you want to capture as many people as you can beforehand, and then be able to know what's happening through them over time. And then the ability to recontact or others allows you to answer some of those questions that you may have as well. Technologically, I think we need to have data sets that are integratable, that are cross-linked, and that we can deduplicate on to enable greater power in those kinds of studies and help us capture, especially when you think about conditions around birth, mom-baby linkages and things like that, that would enable you to understand the prenatal environment, for instance, as that one example, better. And there would be similar examples for other kinds of conditions over the life course as well.
Great, thank you. It's complex, and we recognize we have to be able to look backwards as well as looking forward. Thank you so much. Do we have other questions, Jomi, before we move on?
Yep, we just had a couple more come in. So first question is the All of Us data geocoded so that it can be linked to neighborhood focused or ecosystem census data like the Neighborhood Risk Index or others?
Correct. Yes, we are. We ask individuals their location. It is geocoded. In the current data set that's released, it's at a higher level. You just have state and EHR data provider. But we will, as we release and build out more features, we'll be able to release more granular information. And in addition, we will centrally link data that we will bring into the data sets as well.
Thanks. And we have time for one last question. So it is All of Us collecting any data on psychological traits that are known to predict health behavior and outcomes, such as patience, risk preference, and the Big Five?
Thank you. So, one of the things we've done with our COVID survey is we asked a number of things beyond, just do you know someone that's been affected by COVID? Have you been affected by COVID? We also asked things about like optimism, resilience, discrimination, depression, anxiety, and those kinds of things as well. So, and we will continue to release more of those kinds of surveys over time. So yes, we are trying to capture a more complete picture of what's happening to individuals.
Thank you. So that's the time we have for questions. So thank you very much for answering that.
Great. Thank you so much, Dr. Denny. We really appreciate you being here and also your leadership. I'd like to now move us to the how-to talks. So we are really fortunate to now turn to Dr. Norrina Allen. She's the director for the Institute of Public Health and Medicine. She specializes in cardiovascular epidemiology with a focus on health services research. Her research interests include the effect of neighborhood and environment on cardiovascular disease, disparities in the quality of care and outcomes, as well as the use of large databases to examine these questions. Welcome, and thanks for being here today, Dr. Allen.
So I am excited to follow Dr. Denny and get into a bit more of the nuts and bolts of how to actually prepare and package the integrated data sets. And I'm going to be specifically talking today about the integration and harmonization of diverse data resources. So the two objectives for this portion of the talk are really to define a common language for integrative data analysis, integrative data sets and their analysis, and to identify key methodologic considerations when you're designing an integrative data project. I'm going to talk about those objectives in the context of two different portions of integrative data research. One is really work that's designed to increase the breadth of the study. So to increase the diversity of a sample size across populations. This is what we generally think of as pooling multiple samples together to get a much larger representative population. But there's a new and growing trend to data linkages that Dr. Denny mentioned to really increase the depth of information we have about any individual within our within our population or our cohort. And so I'm going to break those two apart to talk about the kind of how-to issues that are associated with each of those study designs. So to start off, first, I'm going to talk about pooling and harmonization. And I think I would start off by saying that integrative data sets, I think, are really the future of EPI. This is really, I think, where we're going. And I'm sure you're all aware that there have been a growing number of publications which are combining multiple cohorts or bringing together multiple samples. As Dr. Denny mentioned, the field of genetics has really been leading the way in this. And that's partially because there's some key advantages to these pooling studies. The first is you obviously get a much larger sample size. So you can examine smaller effect sizes, you can examine rare outcomes without having to do a case control study, and you can really disentangle phenotypes that may be more heterogeneous than we once thought. An example of that might be heart failure with preserved ejection fraction. It's increasingly understood that there's a variety of underlying phenotypes there, which each have different pathophysiologies that lead to HFpEF. In addition, by pooling multiple cohorts together, you get a more diverse population. So you can really represent the breadth and uniqueness of different groups across the country or across the world, and you can really enable yourself to look at subgroup analyses to look at similarities or differences across those groups. And lastly, I think you also have this ability to follow individuals across the lifespan. So you can really combine cohorts in a way that allows you to get a full picture of a life course perspective for any disease and examine different trajectories in that life course perspective. And that last piece is really my particular research interest. And so if we're just taking that as an example, thinking about the life course perspective in just healthy aging, we know that the trajectory of aging actually begins very early in life, and some might argue even prenatally, and that the early differences may be influencing later patterns and disparities in healthy aging. To date, we've mostly had information from specific cohorts on one or two of these major life stages. So we've got childhood cohorts, we've got adult cohorts, and we've got older age cohorts. And more recently, we've had studies like Framingham, for example, where they've actually gone and extended across two different age periods. What we're particularly interested, though, is this full life course aspect. Can we actually understand the origins of disease and also see them play out into older ages so that we can understand these differences in trajectories? So just as an example, we have a proposal for a life course consortium. We've been working with cohorts across the world to really create this life course perspective. And what you can see here is there's on the top are the adult cohorts and on the bottom are the childhood cohorts. The x-axis is age in years. And what we wanted to do is we wanted to pool cohorts in order to allow us to have information across the full spectrum of age, from childhood all the way to older age. And by pooling these cohorts, we can actually achieve course perspective on how early life influences may impact later life trajectories of health. Obviously, when you're combining multiple cohorts, a large number of cohorts, there's a lot of work that has to be done. Each of these cohorts was designed and the data was collected to be independent of each other. And we're now post hoc trying to put them together into one larger sample. And so that requires a lot of harmonization. So by harmonization, what I am referring to is efforts to combine the data from different sources and from different users or different studies. So with its own unique protocol and its own unique platforms. And what we're trying to do is combine them in a way that we can get an usable picture of what the life course of a disease is. So when you're thinking about harmonizing data, we have a couple key considerations that we have to take into account. The first question is, can we standardize and optimize the harmonization process, both scientifically but also organizationally? And we want to maintain rigor while maintaining the costs that we actually have available to us. What measures can we use to determine how successful our harmonization has been? obviously we can't just throw everything together and hope that it was measured in the same way. We have to have some way to evaluate the harmonization process. What's the minimal and optimal level of harmonization that's needed to ensure correct inferences? And also what levels and types of documentation are needed from collaborating studies to ensure maximum efficiency of the harmonization process. So obviously you can get all the protocols, you can get their data dictionaries, and then begins the work of actually harmonizing. So if we just look at some of the adult cohorts, these are the cohorts at the top. You can see that each one has had a variety of different examination cycles at different time periods over the course of the past 30 years. For example, this is the 3-city study. You can see their baseline was about 1999, and they had 3 follow-ups going through 2012. At the end here is Whitehall, and you can see again they started much earlier and have had many more in-person exams. So when you want to start off with harmonization, you have to start off with truly understanding the data. So it's not only the structure of the data, but at each of those exams, different information was collected in different cohorts. This is just an example of cardiovascular risk factors, and we were just trying to track which of those risk factors was collected in which study and at which time point. And so you can see that it gets very complicated very quickly, and maintaining rigorous documentation is essential for harmonization process. When we talk about harmonization, what we're talking about is a variety of different approaches. So we use different methods to actually ensure the harmonization of different types of variables based on what we can do and what we need to do to actually make scientific contributions. So what we most often see is algorithmic transformation. So that's really just kind of lining up categories. Often we see this when we're talking about the highest level of education or race, ethnicity. We're just trying to align categories that are combinable. We also often see simple calibration models and cardiovascular risk factors. This is making sure really that the units are on the same scale, that if we're going from pounds to kilos, we'll all have a consistent metric. We then often see standardizations where we see z-scores. And so we're just trying to make sure that the distribution of those items is consistent. With z-scores, we don't often have a way to back translate the absolute value. What we're doing is looking at the range and distribution. So each person is compared to other people in their same cohort across the distribution. Less often we see more latent variable constructs or multiple imputation models where we're trying to actually use statistical modeling techniques to actually harmonize data that may have been collected in very different ways, but maybe all trying to get to the same underlying latent variable, or may have been collected in different scales, but we have some way to overlap those 2 scales. Maybe one study collected both of those scales, and we can kind of impute across cohorts. Each of these methods is used for different types of variables and in different settings. When we apply a harmonization, we then have to evaluate how well our harmonization, whether we succeeded. And often people think you succeed in harmonization if all of the cohorts have similar values of that variable across cohorts. But what actually we need to be aware of is that that's not actually the metric of success. We expect there to be differences in many of the risk factors and many variables across cohorts, but we need to understand why those differences may exist, and we need to make sure that those differences are true differences and that they don't just represent methodologic differences across the cohorts. So in one example, we were harmonizing diet, which is actually very complicated across cohorts because it's not collected in any standardized way. And we did multiple different methods in parallel, or in sequence, I should say, to actually harmonize diet. So we had to align the types of foods that each study collected. We then had to align the serving sizes that each study collected, and then we had to do imputation. But you can see in this subset of the cohorts, what we calculated was the alternate healthy eating index. This is paneled by each of the cohorts: Eric, Cardia, CHS, Framingham, Offspring, Jackson, Mesa, and WHI. It's separated by gender for women and men. And you can see that we were looking at the means and medians. So we were comparing the distributions across the cohorts. And the distribution was similar. The means and medians were similar across the cohorts, but you'll notice there were differences. We do expect there to be cohorts that had maybe healthier eating patterns than other cohorts that may have had poorer eating patterns. But at the end of the day, the success of harmonization is that we acknowledge that there's differences in the cohorts that are not due to methodology, they're due to actual cohort differences. And so a lot of harmonization, again, is examining why you get the results you get and really understanding what their differences are. So when we're thinking about harmonizing, at the end of the day, there's two strategies for actually doing the analysis in these pooled cohorts. And so I just wanted to highlight that we can do a true pooling or combining the data together into a single cohort. This is often called integrative data analysis. It's essentially bringing all of the different cohorts together into one large cohort, sometimes a synthetic cohort, and doing the analysis as a single sample. Adjusting for or stratifying by the cohort itself. The advantages of doing it this way are that it allows you to utilize a wide variety of statistical methodologies, but it does assume that all those populations are this part of the same underlying sample. And so there's some questions about really understanding why there may be differences across cohorts, and a lot of investigation has to go into that. Another option is more federated or meta-analytic methods, in which the analyses are done on the harmonized data sets in parallel or sequentially across the cohorts. An advantage of this is that data can be harmonized in different sites around the world and then analyzed in its own home institution, and the outcomes, the effect sizes can then be meta-analyzed together to create a larger understanding. But you must have appropriate meta-analytic techniques to be able to create that single pooled estimate if it's appropriate. And not all of our statistical methods have that meta-analytic method to combine and effect sizes. An advantage of this meta-analytic method is it intrinsically takes into account population differences. And so it's very clearly set up to understand whether there are certain samples or populations that may be different than others. So that's the first part, really thinking about pooling cohorts across a wide variety of individuals. There's another type, and that's what I was talking about in depth, is linking diverse data sources to more clearly phenotype and the individuals within your cohort. And so Dr. Denny mentioned this a little bit when he was talking about the All of Us study. So it's increasingly clear that there's a wide variety of information that's collected about people within our cohorts, and that if we brought that information together from a wide variety of sources, we might get a better picture of what their exposure and their risk and their outcomes is. And so this is just a subset of a wide variety of information you might be interested in knowing about any single cohort participant. But we can get information from disease registries, claims data, electronic health records data, pharmacy data, wearable devices. We use administrative mortality data, labs, biomarkers, social determinants of health, and there's a wide variety of data sources that we could add to this picture of what each individual's life looks like. This is often thought about in terms of deep phenotyping on the omics side of things. There's similar digital phenotyping and a wide variety of exposome. So really understanding the diversity and the complexity of the environment and physiology of each individual. And in order to do that, we have to bring data from across different sources together to create this unique picture of each person. Importantly, though, when we're linking these diverse data resources to really expand our understanding of each cohort participant's phenotype, we have to consider a couple additional issues. And one is identifying and linking while protecting the privacy of that person. We can't be sending around that cohort participant's name and social security number to different places in order to link them. We also need to have a common ontology and really be concerned about data stewardship. So I'm going to just talk about one method that we use often to preserve privacy, and we use PPL, like a privacy protected linkage. And the advantages of privacy protected linkages are that one site, in this case, let's say site A, let's say this is a cohort and we want to link with electronic medical records, site B, we don't have to share our participants' information, and the hospital doesn't have to share the patient's information with each other. We may be at different sites, we may be across the country. What we can do is we can each identify that participant or patient's PPL number or hash ID, and then that hash ID is the only thing that gets shared with the linking center of site C. So that hash ID is unique in that if I put the same identifiers in, the same name, the same address, the same social security number, I will always get that same hash ID out. And there's more complexity to that, to be sure, but essentially that's the underlying concept. And so that if site A puts that same information in and site B puts that information in, they will both end up with the same hash ID. And that hash ID is privacy protecting, but it allows us to link across sites using a kind of honest broker in this linkage center. And then we can then strip all the identifiers out and link the data based on the hash ID, strip those out, and link by our study ID. And that allows us to preserve the privacy of our participants, as well as the privacy within their healthcare records, but allows us to identify a one-to-one linkage of a cohort participant with their electronic medical records. And these PPLs are used across a wide variety of settings, and increasingly they are allowing us to identify data for our participants without actually sharing any identifiable information. The next big concern is really having a common ontology or a shared data model. And so having a common ontology really means that we have a shared vocabulary or taxonomy of the data. And we're explicitly defining how these different data sources are related to each other. So what do I mean by that? Well, many of you may have heard of OMOP. So OMOP is a common data model, observational common data model, and it's often used in EHR records, but the same theory can be applied across a wide variety of data sources. So the OMOP common data model is designed to enable standardized analysis across different data modalities, really allowing for collaborative research and data sharing. So if everyone agrees on this common data model, then we have essentially kind of pre-harmonized our data and we can share it across data sources. An important part of the OMOP model as well is that there's a relationship across each of these variables. So we understand the visits are within the individual who's within the provider, which is within the care site. So these structural relationships are preserved and very clear, and it can be expanded out much broader than EHR, but to understand when we're incorporating mobile platforms, when we're incorporating a wide variety of information from different sources, how they relate to each other and how they relate back to the study participants. One example of that we did a couple of years ago was really linking our cohort with a large electronic health record data set, which was called HealthLink. So it provided electronic medical records for individuals across the city of Chicago. About 3 million unique individuals were identified in HealthLink. And what we were interested in is pulling our MESA participants and understanding their healthcare encounters and their routine care within the electronic medical records. We do validate events and we have medical records for events, but we didn't know anything about their routine encounters and their labs and their vitals from their outpatient visits. And so we linked the participants to the HealthLink, and you can see kind of the flow and all the issues that came up. Like I said, this was a couple years ago, and we're learning quickly about how to link these cohorts with electronic medical records. But I think it identifies some key considerations in really understanding the underlying sample. So not all of our MESA participants sought care within HealthLink hospitals. And then even within those, some of them didn't have all their labs and vitals. And so you can see we started to have to whittle away, and we looked at the reasons for those exclusions and how we could supplement them. But at the same time, we had to have this common ontology. We had to be able to say blood pressure in MESA is similar to blood pressure within the EHR system and how they related to each other over time. And lastly, I'll just end with the fact that with all of this data, both pooling and with unique data linkages, really the data stewardship becomes an important aspect of what our job is as researchers. There's a wide variety of information that's being collected on our diverse group of people across the world. And our concern as researchers really needs to be protecting and representing the concerns of others. So when we're pooling data across cohorts, we're very much concerned about the fact that we are doing this in a collaborative environment. We are representing the concerns of each of the cohorts, and it's done with their help and collaboration and their willing involvement. But I think even more importantly, it's also done with the participation of the individuals themselves, the participants, the patients. Our goal is to protect their confidentiality, to protect their data, and to really act with privacy and respect towards the information that they've willingly given us to pursue our research. And so that's where I'll end, and really that trust is given to us, and we need to live up to it in all that we do with data pooling. Thank you very much.
We're going to move into our last how-to talk. The last how-to talk is presented by Dr. Nick Tatonetti. He is an associate professor in biomedical informatics at Columbia University. His research interests include translational bioinformatics, machine learning, observational data mining, combinatorial drug design, emergent biology, genetic networks, and network analysis, as well as clinical data analyses. He's going to talk about mining massive observational data. Dr. Tatonetti.
Hello, everyone. I am Nicholas Tatonetti. I'm an associate professor of biomedical informatics at Columbia University, and I spend my time thinking about how to use massive, large observational data sets to make scientific discoveries. And today we're going to be talking about something called data mining, which is essential for that process. Observations are the starting point of all discoveries. I love starting with this slide. You can see this figure over here. And this is a scanned page out of Darwin's notebook. And I love this because at the top right left corner there, he writes, "I think," and then he draws this beautiful diagram. And if you stare at this diagram, you can start to see the connections that Darwin is making between geography and phenotype that, of course, would lead to the theory of evolution. And this is not just true in biology, but in a field that I study very deeply in adverse events, drug adverse events, or pharmacovigilance. In fact, there are these two scientists, McBride and Lentz, from opposite sides of the world, were making observations of their patients. They were noticing that the pregnant women who they were prescribing thalidomide seemed to have more birth defects than other women. This was a really surprising result, and the birth defects are very severe. But these observations led to the discovery and validation of thalidomide as a potent teratogen, in fact, kicking off modern pharmacovigilance and adverse drug effect studying as we know it. The tools in how we are making observations are advancing. At first, we started with our human senses. We would go out into the world like Darwin sailing on the HMS Beagle to the Galapagos Islands and look at things and taste them and see them and write them down in our notebooks. And we started augmenting these abilities with microscopes and telescopes so that we could see farther than our eyes would naturally let us see, or hear things that we otherwise could not hear. In addition to that, we started to invent different biochemical or screening technologies, different ways to augment our senses in unnatural ways. So chemical screening, high throughput screening, microarrays, and next generation sequencing are all examples of coming up with new senses, using the discoveries that we've made to collect observations at scales that we've never seen before. So when Darwin and McBride and Lentz were going out into the world and collecting observations, they were collecting bytes or maybe kilobytes worth of data. But today's scientists are collecting petabytes worth of data with every sequencing run. It's an amount of information that's very difficult to capture in just one person's and one scientist's mind. So how do we deal with that? And in fact, it doesn't just stop there. Technology is making it easier to collect massive clinical data sets, observations at a global scale about how patients are treated, what drugs they are taking, and what those outcomes might be. In fact, over 98% of hospitals in the United States are using electronic health records, collecting observations on every patient that walks through the door. Those observations, each of them could lead to new discoveries. Just to give you an idea of the scope and the scale of the type of data that's available to observational clinical research, this is an example from just one hospital, Columbia University Medical Center, where we have over 6.5 million patients. And for these patients, we have an incredibly rich data set of prescription orders, of clinical diagnoses, of procedures and surgeries that occur, and every lab test that gets run. Every time your height gets measured, or your weight gets measured, or blood is drawn for tests. And in fact, on top of that really rich structured data, there's also the unstructured data, the notes that are written about patients, how this patient is being cared for, what is the treatment plan, what is the prognosis for this particular patient. All of this kind of represents a particular patient and is incredibly detailed. And then if you multiply that by 6.5 million, you have this massive data set. But that's just one hospital. Across the world, there are thousands and thousands of hospitals just like ours at Columbia University. In fact, the average patient generates about 80 megabytes of data per year. And that's even accounting for the fact that most of us only go once or twice. There's a problem with using these data though, these observational data sets that look so rich and so promising, that keeps us from making huge discoveries. And that has to do with missingness and bias. And to exemplify this, I like to tell this story about a red bird and a blue bird. So I'm here in my apartment in Manhattan, a few blocks away from Central Park. And so I often go through Central Park and I like watching all the birds. And I've noticed that there are red birds and blue birds in this park. And so we're curious. So we want to go out there and figure out what's the relative population of red birds to blue birds in Central Park. Seems like a fairly reasonable question. So we grab a notebook and we walk to Central Park and we take a hike. And every time we see a blue bird, we make a mark on the right. And every time we see a red bird, we make a mark on the left. Then we get back to the apartment, to our home laboratory, and we analyze those results. And what we observed was that we observed 5 times as many blue birds as red birds in Central Park. Now, what does this tell you about the question that we asked? What is the proportion of the population of blue birds to red birds? Absolutely nothing, because now I'm going to tell you something else, which is that bluebirds, you might be able to tell from this graphic, are really friendly birds. When they hear people come into Central Park, they know they drop food all over the place. They're super excited to go chirp at them, fly at them, and try and get some of those treats. But redbirds are completely the opposite. They just want to be left alone. They don't like people. When they hear people come into Central Park, they hide, they flutter and fly away, and they hide in the North Woods. So the behaviors of these birds has actually invalidated your observational data set, making it impossible to answer the very simple question about the relative populations of these birds. And this is not just a hypothetical example. This is happening every day in the hospital records. The behaviors of patients, the behaviors of doctors and nurses and other care practitioners changes what we can observe in the hospital records, making it often invalid or impossible to answer seemingly very simple questions. Examples of these type of things include which diagnosis codes. We can observe diagnosis codes. We can observe lab values and vitals and surgeries, all those data I was showing you before. But we don't know the environmental triggers, the environmental exposures for the patient. We don't know what other hospitals they might have gone to, and we don't know the peculiar treatment decisions or the information that might have gone into that is not captured by those databases. I want to give you a very specific example. There's a series of scores that you can use to evaluate the risk to a patient who has pneumonia. One of them is the CURB, another one's called the FINE score. Now, we've attempted to use these scores by automatically generating these scores from the electronic health record data. In fact, this is a paper published by a colleague of mine that shows that when you apply this score to the electronic health record data to run this automatically, you find something very nonsensical. You find people that are, according to the score, at the lowest risk actually have high mortality. These people should be totally fine. And if you apply the score has been validated. So this doesn't just make any sense. What's happening here is that these patients who died very quickly upon admission or upon arrival, simply don't have very much data in their records. And the fact that they don't have much data makes it look like they're a healthy patient. So these types of biases can totally confound very simple analyses, making the interpretation impossible or very difficult. So data mining is about taking these things into account, about designing automated algorithms that can dive into data sets, account for some of these issues of missingness and bias to generate good hypotheses. And remember, good hypotheses are testable. They make predictions that are testable, and they address these issues of bias and confounding, at least to some extent, so you can do further analysis. And then also, since we have all of these data, we have a new opportunity. Instead of asking one question of one data set, we can ask that same question of totally different data sets using different analytical strategies. And if all of those different strategies tell us the same thing, then that hypothesis is much more likely than others that don't have that same level of evidence. And I like to think about how to use massive observational clinical data sets to make scientific discoveries. Today, we're going to be talking about how to use data mining to identify previously unknown and dangerous drug-drug interactions that cause a heart arrhythmia called acquired long QT syndrome. Now, acquired long QT syndrome is a dangerous drug-induced tachycardia, a heart arrhythmia that can occur when patients are exposed to certain medications. And because of the importance of this potential adverse event, the FDA mandates that every single drug used individually is tested before clinical trials. If a drug is shown to have an effect on the important ion channel that governs this adverse event, then it almost never goes into clinical trials or becomes available for use. But no such mandate exists for drug-drug interactions. In fact, we must rely on observational data of patients already taking these medications together, data, and then reporting potential side effects in order to identify these drug-drug interactions. But these databases are full of potential false positives due to issues with missingness, bias, and confounding. In order to trust the type of hypotheses that come out of data mining such a database, we take an integrative approach. We don't rely on just one analysis and one data set to tell us that a hypothesis might be true. We integrate multiple orthogonal databases and multiple analytical techniques together to improve that hypothesis, to bolster that hypothesis. In fact, the more different they are, the better we feel about those hypotheses that come out of that data mining. In this analysis today, I'm going to show you how we data mine the FDA's adverse event reporting system, a side effect database, to identify putative drug-drug interactions. How we then shifted and used an independent database called the electronic health records to corroborate that hypothesis, and how we use prospective experiments in human cell line to validate this drug-drug interaction is occurring. We use an algorithm for data mining called latent signal detection. It's an algorithm we developed to identify adverse events that are occurring, even if there's no direct evidence of them available. In fact, it's inspired by how scientists hunt for planets. Instead of looking for the planet directly, which would be almost impossible with our technology, what we do is we look for the effect of a planet on the star, and that we can observe. In a similar way, what we're going to do is not look for the effect directly, but look for its indirect effects. If a drug-drug interaction is happening, it means it must be causing not only the effect we're after, but many other observable effects that we can then model. In this algorithm, we hypothesize that there is a latent, unmeasured, severe adverse event lying under the level of detection. We can't see it directly, but that this latent effect has certain signs or symptoms, measured minor effects that we can model. We're going to model these effects using supervised machine learning, and then we're going to take those effects and build an effect profile. Using this profile, we can then scan through all possible drug pairs in these databases and identify pairs that match this profile most closely. These will be our putative drug-drug interactions. We built such a model for acquired long QT syndrome to look for drug-drug interactions causing this severe and dangerous effect. And these are a couple of the signs and symptoms that are in the model, the more minor observable effects: bradycardia, atrial fibrillation, and tachycardia, among some others. Our top prediction when mining the FDA's adverse event reporting system using this effect profile was between ceftriaxone and lansoprazole. Ceftriaxone is a very common inpatient antibiotic. In fact, it's a first-line antibiotic at our hospital here at Columbia University. And lansoprazole is a proton pump inhibitor. It's one of the most used drugs in the world. You can actually buy it over the counter as Prevacid, among other brand names. Now, an interaction between them, especially one that causes a dangerous heart arrhythmia, could have very severe consequences. This is what the data mining saw when it scanned the databases. This is the signal that it saw that triggered to the algorithm that this might be causing acquired long QT syndrome between ceftriaxone and lansoprazole. Now, we went one step further and we asked the algorithm to make a prediction of a negative control so that we can walk both of these potential drug-drug interactions through the pipeline and feel more confident in our prediction if our negative control doesn't have the same effects. So the algorithm identified a negative control, also a cephalosporin antibiotic in combination with lansoprazole, and it does not have the same triggers, it does not have the same signs and symptoms. Then we go to the independent analysis in the electronic health records. And what we found there was that patients that were exposed to the combination of ceftriaxone and lansoprazole had 10 millisecond longer QT intervals than patients who were exposed to just one of the drugs, an effect that we did not see in the combination of cefuroxime and lansoprazole. In addition, we see a significant proportion of patients have dangerously long QT intervals over 500 milliseconds that would raise concern for this potential tachycardia to develop. Now it's time that we have these two sources of evidence to go to our prospective experiments to validate that this interaction is occurring in cell lines. So we take these cells that are overexpressing an important ion channel called the hERG channel, and we expose them to a control. We expose them to each drug individually, ceftriaxone and lansoprazole, and then we expose them to the combination of ceftriaxone and lansoprazole. What we find is that when these drugs are given in combination, they cause a dose-dependent effect on this important ion channel, an effect that you don't see in the predicted negative control. And in fact, if you put this back into a model to predict what would happen to patients who are exposed to this combination of drugs, you would expect to see a 10 millisecond longer QT interval in those patients, exactly what we saw in the electronic health records. So in this study, I've shown you how we can use observational data sets. We can mine them for hypotheses, that we don't trust those hypotheses outright, that we corroborate them with independent analyses of electronic health records, and then finally that we validate them prospectively using a completely different technique to prove that this interaction is actually happening. Thank you.
Well, I just think that was incredible. And I think what Dr. Tatonetti leaves us with is when we're using these large integrated data sets, we have to be really cautious. We want to make sure we actually are finding the signal through the noise. We have to identify bias. We have to really elevate the rigor of our assumptions, our biases, our missingness. If you are utilizing that technique, it's about mining for hypotheses, but then corroborating and then validating. So we leave you there. We thank the more than 60 of you who have stayed extra, and we hope that you have a great day. Thanks, everybody.